Welcome back, students. In this session, you'll be learning about digestion and absorption of carbohydrates. I know it looks very simple. It might be something which you have learned from before, but there are many questions that come from digestion and absorption, and you shouldn't want to miss on that. So hence I'm going to deal with this digestion and absorption of carbohydrates, which you might have forgotten also. So what is digestion? We all know that digestion is the process whereby big macromolecules are converted into small macromolecules. Till there you call it as digestion. So why do we convert it into smaller molecules? So that they can be absorbed and they can be utilized by the body. So we'll go on to the dietary sources of carbohydrates. This is something which you are doing daily, something which you are eating daily. Every meal in your house will be containing carbohydrates. So let us talk about this from that viewpoint also. Which are the dietary sources and what is their composition? Now, which carbohydrate is there, which is the typical source, and what is its composition? Again, I am giving it to you in a biochemical perspective. Now, one important carbohydrate is amylopectin. Where is it found? It is a component of starch found in potatoes, rice, corn, bread, etc. And please note the composition. It is made up of glucose and glucose with 1,4 and 1,6 branches. Amylose is another component of starch, again found in all these, but it is having only 1,4 linkages. Glycogen, do you take it in your diet? Yes, even though it is present in your liver and muscle, but if you are a non-vegetarian, definitely glycogen is going to be a part of your diet. It is present in the liver and the muscle. So glycogen is also a part of your diet. It is again made up of alpha glucose with both 1,6 and 1,4 linkages. Another important carbohydrate, source of carbohydrate is sucrose. How do you take it? Table sugar, desserts, ice creams, anything you eat, gulab jamun you eat, you are eating sucrose. So, it is again made up of glucose and fructose in 1,4 linkage. Please remember, it is beta fructose in alpha 1 beta 4 linkage. Lactose is another carbohydrate that you take. How do you take it? What are the sources? Milk and milk products. Any milk products. You are taking paneer, you are eating cottage cheese, you are eating parmesan cheese, whatever cheese you are eating or whatever is a rasgulla you are eating, if it is a milk or a milk product, then it will definitely contain lactose, which is made up of galactose and glucose. Now fructose is present in fruit and honey. Glucose is present in fruits and honey as well. These are all monosaccharides. That's why I have not told you about its composition. Cellulose is present in plant cell walls. You are eating palak, you are eating any of the bhajis. So what is present in these plants? They have cell walls. So when the cell wall is there, what is the carbohydrate that is present? It is cellulose. It is again made up of glucose in the beta 1,4 linkage, many number of glucose. Trehalose. Trehalose is another carbohydrate which is present in young mushrooms. So mushroom is a part of your diet. You may be eating trehalose. Again, this is made up of glucose and glucose in 1,1 linkage. What is happening to all that you are eating? What happens? First you put it in the mouth. So what is happening? There is the saliva. So carbohydrate digestion starts in the mouth with the enzyme salivary alpha amylase. This is an MCQ. To which class does it belong? As I have told to you repeatedly, they all belong to the class 3 hydrolases. So it belongs to class 3 hydrolase, which uses water and breaks up the glycosidic bonds. So which sort of bonds does it break? It is an endoglucosidase. Endo means within the molecule. So it is an endoglucosidase. So it randomly breaks whichever glycosidic bond it wants. So you have a huge carbohydrate, it breaks wherever it wants. So it is an endoglucosidase. But it is very specific for alpha 1,4 glycosidic linkages. So it breaks down a huge molecule of starch into what? Wherever it breaks, so it will result in the formation. And remember, it cannot act on 1,6. So it results in the formation of limit dextrins, or we call it as dextrins, maltotriose and maltose are formed. It cannot act in the acid pH of the stomach. Minute you swallow your food, a salivary alpha amylase action is going to stop. And also remember that the time available is very much limited for salivary alpha amylase. In the stomach, no digestion. HCl will just inhibit the action of salivary amylase. In the duodenum, so now the food has reached the duodenum. What happens? Pancreatic secretion comes. What does the pancreas secrete? It secretes not only bicarbonate to make the pH alkaline, but it also has an important enzyme, pancreatic alpha amylase. This is also similar. It is an isoenzyme of salivary amylase. It also hydrolyzes starch and glycogen present in the muscles of different non-vegetarian foods, forming maltose, which is a disaccharide, maltotriose, which is a trisaccharide, and oligosaccharides called as limit extremes, 4 to 9 glucose with 1 or more alpha-1,6 glycosidic linkages. It also produces isomaltose with 2 glucose with alpha-1,6 linkages. Alpha amylase acts only on polymers containing glucose and only on alpha-1,4 glycosidic linkages. Look at the specificity of this. Alpha amylase only on glucose and alpha-1,4 glycosidic linkage. So can it act on sucrose? No. Can it act on lactose? No. It cannot act on these two substances. So please remember that it is very specific only for where there is alpha-1,4 glucose to glucose linkage. That is where it can act, and it cannot act beyond that. And it cannot act on other substances. Now in the intestine, what happens? Still in the intestine, duodenum only, the food is going down. There are something else which is required. So we need the salivary gland to secrete salivary alpha amylase. We require the pancreas to secrete pancreatic amylase. Other than that, we need some other enzymes. The digestion of carbohydrates is still not over. So the oligo, tri and disaccharides are converted to monosaccharides. Please remember that it is only the monosaccharides which can be absorbed by the intestinal mucosal cells into the portal vein. Rest of this other disaccharides or trisaccharides or oligosaccharides are not absorbed. So there are certain enzymes which are going to convert all this into simpler monosaccharides. They are called as glycosidases, which are attached to the membrane in the brush border of the absorptive cell. Please remember that the small intestine, everything has a lot of villi are there. These villi of the each intestinal mucosal cell will increase the absorption surface of the intestinal mucosal cell. But at the same time, these are the ones which are secreting the enzyme. Hence, these are also called as the brush border enzyme. Now, the brush border, why do we call it brush border? As I said, these are present in the intestinal mucosal cell, which has a lot of villi in it. Which are the brush border enzymes? We include glucoamylase, isomaltase, maltase, sucrase, lactase and trehalase. And some of these enzymes are complex together like the sucrase isomaltase complex. We go on to the actions. I have been telling you the specificity and the substrate and the product. This is a huge table which gives you all of this together. Now salivary amylase, alpha-1,4, starch and glycogen to alpha-dextrin, maltose and maltotriose. Pancreas, pancreatic amylase, alpha-1,4 again, starch and glycogen to alpha-dextrin, maltose, maltotriose. There is no difference in their action except that one of it requires chloride ions. Glucoamylase is an exoglucosidase. It acts on 1,4 from the non-reducing end, and the substrate is amylose, product is glucose. Isomaltase, what does the name suggest to you? Isomaltase will act on isomaltose. What is isomaltose? Glucose in 1,6 linkage. So isomaltose and alpha-dextrins are converted into glucose. Maltase, what does it act on? Maltose. It is again 1,4 linkage. So maltose and maltotriose are converted to glucose. Sucrase, where what does it convert? Its specificity is alpha glucose and substrate is sucrose, so it will convert glucose into fructose, sucrose into glucose plus fructose. Lactase will act on specificity is beta galactose, so lactose is converted into glucose plus galactose. Trehalase will act on alpha-1,1 linkage, so it will act on trehalose and convert it into glucose. So these are the specificity and the different enzyme. which can be formed in this. Now all the monosaccharides are formed. They have been digested. A complex molecule has been converted into its component monosaccharides. So what is the next procedure? These have to be absorbed. So how is the absorption going to take place? Again, as I said, it all depends upon the absorption surface is very huge in the intestinal mucosal cell. Why? It contains a lot of villi. Now the transport across the intestinal cell from the lumen into the portal vein or into the blood, we require certain things are happening out there. So the transport of the products, monosaccharides, that is glucose, galactose and fructose into the enterocyte. Now what is happening here? Now you have the lumen over here. I'm just going to show you one transporter which is present at the lumen, and that is, this is a sodium dependent glucose transporter. This is SGLT. So this is, not only does it absorb glucose, but along with that it will absorb sodium also. This is a type of secondary active transport. So along with glucose, sodium is also absorbed. That is the importance of sodium glucose transporter 1. So this is a sodium dependent glucose transporter which is specific for not only glucose but also for galactose. Both these are transported with the help of a secondary active transport, glucose plus galactose. What about fructose? Remember the glucose transporter 5. GLUT5 is responsible for the transport of, I'm just showing you simply a schematic diagram. So glucose transporter, we call it glucose transporter 5, GLUT5. GLUT5 is responsible for, very specific for fructose, wherever it is, whether it is in the kidneys or in the intestinal mucosal cell, fructose is transported via glucose transporter 5, which is not dependent on sodium. GLUT5 is also sodium independent. Please remember that sodium independent glucose transporter is there for both fructose and galactose, but for both fructose and glucose, galactose always requires sodium dependent glucose transporter. Now once these all have entered the enterocyte, they have to go out. And how do they go out? It is a simple facilitated diffusion, and this is by glucose transporter 2, GLUT2. GLUT2 is got nothing to do with any sodium or anything. All these are transported via glucose transporter 2. And MCQ question: does it require sodium? No. Does it require energy? No. Is it facilitated? Yes. Is it follows the ping pong mechanism? Yes. These all are follow the ping pong mechanism and helps the transport of glucose and galactose and fructose into the blood. So glucose GLUT2 is a facilitated transporter for glucose, galactose and fructose to leave the mucosal cell and to enter the portal circulation. Is absorption over? Yes. This is a simple way by which absorption is done. Now I'll give you an important clinical feature. Digestion is over, absorption is over. One important thing that is to be known regarding all this, that is known as glycemic index. What is glycemic index? It is a measure of the rate at which a carbohydrate in the food will enter the bloodstream as glucose. What is the rate at which any carbohydrate will enter into the bloodstream as glucose? So, a food with a high glycemic index, what does it mean? Will release glucose into the blood quickly. More the glycemic index, more it will release glucose quickly into the blood and cause the blood sugar levels to rise rapidly. The high glycemic index means it will raise the blood glucose very rapidly. What do you think will be? Can you give an example for this? So you have heard of any sugar, for example. If you are taking Glucon-D or any of those, which you need for instant energy. So how is that instant? There is no digestion involved in Glucon-D. All that is glucose, pure glucose. Pure glucose is absorbed pure glucose. So minute you take it, within few minutes it will be go into the intestine. It will be absorbed and the blood glucose level rises rapidly. So glucon D or things like that. Pure glucose will have high glycemic index. What about low glycemic index? They release glucose more steadily and they keep the blood sugar under control. They won't cause high increases in blood sugar. So when is this important? Again, we need this to know about this glycemic index in case of diabetes mellitus, what diet to be given. So the glycemic index, how do we know about the glycemic index of different foods? This is assessed by the what is called as the glucose tolerance test. The glycemic response after the particular diet. You are eating the idlis, you are eating chapatis. What will be the glycemic index of idli or of chapati or of rice or of any chicken? What is the glycemic index? We can do it by doing the glucose tolerance test and usually comparing it with a reference meal. The reference meal is glucose itself. Give glucose, see for the blood glucose level. Give this food and see for the blood glucose level. So pure glucose has 100% glycemic index ranking table and all other foods are ranked in relation to glucose. I am giving some examples here. Glucose is 100%. Apple has a glycemic index of 39%. Bread is 69%. Rice is 72%. Potato 70%. Banana 62%. Sucrose 59%. Oatmeal 49%. So what does it tell you? Higher the glycemic index, worse it is for a diabetic, where lower glycemic index is good for a diabetic patient. So you look at all this and you can prescribe your diet for a diabetic. So you can't give glucose, you can give apple, you can give oatmeal maybe, and then depending on the his requirement you go for bread. You can, but look at rice and potato, look at their high glycemic indexes compared to even sugar 59%. So banana also is, all these are there. Apple has one of the least glycemic index. Now low glycemic index diets are helpful in weight control, not only in diabetics but also in weight control and in improving diabetes control. Body sensitivity to insulin is also maintained and thereby it helps to associate it and thereby associated to reduce the risk of developing diabetes and heart diseases. Basically, what it means is low glycemic index foods are good for us, healthy for us. Now, one more important concept in digestion and absorption is dietary fiber. One we learnt about glycemic index, the other is the dietary fibers. What are these? Now, complex mixture of plant materials that are resistant to breakdown by the human digestive system. The human digestive system cannot digest these carbohydrates. So, they are no use. Yes, they are still useful. We all know about it. These are the undigestible carbohydrates. And remember, there are two types, water insoluble and water soluble. Water insoluble is present in different food items like whole wheat, vegetables, wheat bran, etc. Whereas water soluble dietary fibers are present in oat bran, barley, beans, lentils, fruits, in so many compounds. Let us be more importance physiological effects because of their water solubility or insolubility. Now, water soluble example are pectins and gums are examples for water soluble dietary fibers. Where are they? Fruits, oats, legumes. Now, where are the physiological effect is when they are water soluble, they decrease the rate of gastric emptying. So, gastric emptying takes place slower. They decrease the rate of sugar uptake. So glycemic index will decrease and decrease serum cholesterol. That is the most important of the water soluble ones. The water insoluble include the cellulose and the hemicellulose and the lignin. Pectin comes as water soluble. Gums are water soluble. Lignin is water insoluble. MCQ. Cellulose is water insoluble. Hemicellulose is water insoluble. Again, vegetable, wheat and grain fibers. What do? Water insoluble ones. They increase the stool bulk. They decrease intestinal transit time, decreases colonic pressure, reduces the risk of colon cancer, and they have a cholesterol lowering effect. Basically, both of them decrease the serum cholesterol and they decrease the glycemic index of a particular food. That is the importance of glycemic index. High fiber is a happy colon. Now I'll just talk to you about certain defects in digestion. So disaccharidase deficiency. This is exhibited as intolerance to one or more disaccharides. Which are the disaccharides? Could be lactose or sucrose. Any of this intolerance could be there. Lactose intolerance arises due to deficiency of the enzyme lactase. And this is more prevalent among Africans and Asians. What are the reasons for lactose intolerance? It could be a genetic defect, or it could be physiologically declining with age, or it could be due to injuries due to the mucosa because of bacterial or viral infection. What will happen if it is not digested? The unabsorbed, undigested lactose, what does it happen to it? It goes down. It goes down into the further into the intestine. What happens? There are intestinal bacteria. They start utilizing this lactose. So what? They will convert this lactose into lactic acid and also further into hydrogen. It will convert them into methane and carbon dioxide. So all these gases and all are produced. So what will happen? It will lead to first of all, it will lead to diarrhea. Why? Because there is an osmotic effect. It brings, glucose has got an osmotic effect. Intestinal bacteria have digested that lactose and converted it partly into lactic acid and glucose. Anaerobic metabolism is taking place. So this glucose is an osmotic effect and it causes diarrhea. It also causes flatulence. Why? There is so much of hydrogen, methane, carbon dioxide that is produced. And whenever there is flatulence, then it will lead to abdominal distension due to the gas production. What is important in all this is they can avoid milk and milk products. And one important test is the xylose absorption test, which is used to test the intestine's ability to absorb monosaccharide. And remember, one test is the stools test positive for reducing substances. Glucose and all are reducing substances. So again we have the somewhat a modified Benedict's test like thing, in which reducing substances in stool is looked out and stools will come positive. The sucrose isomaltose malabsorption syndrome, glucose galactose malabsorption syndrome are rare. Most common is lactose and lactose intolerance. With this, we come to the end of digestion and absorption of carbohydrates. Take home message is high fiber, happy colon. So take a good dietary fiber rich with low glycemic index food, which is beneficial to everybody, whether it is to weight loss or to control diabetes. Thank you.